Hello,
Hello.
how are you?
Pretty good. Yeah.
Good.
Mm,
What's going on?
No, well, I think I've persuaded Professor Sharif that he can get a student and do the experiment on salamander eggs. Like, he wasn't sure that I should be in the lab doing this, and that's fine. Like, I would like to see a student do this because then it would give them experience with this equipment. And I don't know. Anyway, hopefully he's going to find someone who would like to image salamander eggs with OCT, optical coherence tomography.
Yeah.
So, yeah. And I finally got, what is it? Workbench to work in ANSYS. So I'm making progress getting started with ANSYS, and that's enough of a project for me right now. Hopefully I'll get my structure on ANSYS and I'll be able to send it to Professor Zhang from the University of Manitoba. He was a supervisor on my master's thesis. And he can see what he thinks about why this structure is so difficult.
Yeah.
I'm going to say it's because it's a tensegrity, but yeah. But he knows how to use ANSYS, and so.
Yeah. All right.
So that's what I'm doing.
Yeah, good. So, Barshan and Morgan, how are you guys doing?
Yeah, hello.
Yes.
Yeah, I'll put in a pull request tonight. I did the final changes and I wrote the commit message and everything.
Okay.
Yeah, tonight I'll put it out.
Okay.
Yeah, I'm just here to see the meeting and everything, see the proceedings. That's
Okay.
all.
Yeah. All right.
Yeah.
Thanks.
Thank you.
All right. So, a number of things to get to. So, first of all, one of our longtime colleagues, Dick Gordon, passed away last week. So, I canceled the meeting, to remember him and, everything. And I'm going to make a longer presentation on his work and our collaboration soon. I'll get it up on YouTube maybe hopefully in a couple weeks. I've been working on the slides, so.
Yeah, he was my supervisor for my first master's degree.
Yeah.
And a colleague.
Yeah.
Yeah.
Yep. So I will talk about some papers today. So this is an interesting paper that came out, I think it was last week or something like that. So it's hot off the presses. This is developmental patterns of the C. elegans neural circuits using community detection. So we've been doing things with the developmental connectome and using network approaches, and they're talking about community detection, and they're looking at the larval worm. So they're looking at the worm from the L1 stage to the L4 stage. So that's what this is. And then the authors, Shubin Wang and colleagues. So this is the abstract. The abstract, C. elegans serves as a prime model for dissecting the developmental trajectories of nervous systems, and indeed it is used oftentimes as one of these small connectomes, where we know all of the cells and we know all the connections. And a lot of times, if we're doing things like the human connectome, we're basically estimating connections between things like voxels or recording sites, and we're building a network from the activity at those sites or in those units. And then we're building a network. We're inferring relationships based on the coincidence of their activation, timing of their act. But in C. elegans, we know every cell, so we can build a map, a physical map of the connections. We can do this in the human connectome too, but it's not at the scale of single cells usually. We can then also build a network that shows activity, electrical activity or chemical activity. And so, we usually, we can use C. elegans as a small connectome. We now have, of course, Drosophila, which is larger, and we know the cells and connections there. But this is, again, a model for disease, a model for things like movement. And then, crucially, we can knock out cells and we can remove them from the circuits or from the networks. And we can see how that changes activity or it changes behavior. Okay, so of course, C. elegans is used as a model. Yet few studies have probed the functional development of its neural connectome. And so this is just doing what I just mentioned, but in development. In this work, we utilize an enhanced version of the Big Clam, which is a community detection algorithm tailored for weighted and directed networks that explicitly capture overlapping modules to delineate neural circuits across C. elegans developmental stages. And they're focused on L1 larvae to adults. And so they're basing this on developmental neural connectome datasets. So I guess they're using a secondary dataset approach, much like what we do. Our findings reveal that neural circuits are initially small and simple during early development, whereas they undergo marked expansion, increased structural complexity, and functional diversification in later phases. Notably, neural circuits emerge progressively. So their finding is that you get neural circuits emerging not all at once, but like in pieces. So what they mean by emerge progressively is that neurons integrate sequentially into functional assemblies, enabling a transition from basic survival-centric behaviors to complex behavioral repertoires. So that means that you get basic components of behaviors and then more elaborate components of behaviors. In contrast to conventional experimental-based approaches, this topological analysis framework facilitates quasi-precise detection of neural circuits with minimal resource consumption. And this is interesting because Jayadratha Gayan, who was a GSOC student, I think it was the year before last or last year, I can't remember which, he's been working on a paper that's somewhat similar to this. On hypergraphs and temporal graphs and things like that. So it's using a different technique. And I would not be surprised if it were the same data set. But this is, we'll see what they come up with here. So the nervous system serves as a central regulatory system, investigating its developmental trajectory from a nascent state to functional maturity is critical for elucidating the formation of its structure and function. Okay. C. elegans, a model organism, features a relatively simple nervous system, making it a good biological model. And so a lot of the development of sort of research directions on this topic have developed. Okay, so this is where we're cited here. Current research on the nervous system development of C. elegans predominantly focuses on observing its neural connectomes across different developmental stages. So this is where a lot of the papers like Whitvliet, and we have a paper on this, Helm, Blevins, and Bassett 2020, and then Kaiser 2017. So thinking about the different developmental stages and things happening at those stages. The observed neural connectome maps, however, merely provide holistic descriptions of the nervous system and fail to analyze the development of neural connectomes from the perspective of neural function formation. So this is where, they're looking at this sort of functional model where you knock out nodes and you see what the result is. Neural circuits constitute the basic functional units of the nervous system. So, if you have something like a movement circuit, you may have 5 or 6 cells that are connected together, and then they control maybe a forward movement or a backward movement. So some cells are recruited for a forward movement, some cells are recruited for a backward movement, and the connections are also important. So if you knock out a cell, you also knock out its connections, and you knock out that part of the circuit. And the question is whether that's essential for some behavior or not. So sometimes if you knock out a single cell, it can significantly disrupt the movement. Sometimes it disrupts the ability to tune the movement, and so forth. And then if we work forwards, we can see that if you think about the network as a structure that forms throughout development, think about the kinds of connections that are gained or lost during development, or maybe like, some sort of defined mutation. So if we have a defined mutant that doesn't have a certain neuron, and there are defined mutants that do have something like that, then you end up with a movement that can't be executed, or a movement that can't be tuned. The same thing happens if you're at an earlier developmental stage and you're moving to a later developmental stage. You gain connections, you gain cells, you gain some function. Okay, so traditional methods for identifying neural circuits requires extensive validation experiments, demanding significant human and resource inputs, albeit with the advantage of precise outcomes. So they introduce here an approach for subaccurate prediction of neural circuits in the nervous system by network community detection. So network community detection is, of course, where we're detecting what we call communities in networks, which are cells that are usually connected together, that go together, that form a single unit. And there are a lot of methods for community detection. They're very intensive algorithmic techniques. So this is just, one of these things that I wish they'll probably expand on it more down here. But I hope that they, kind of give us a good framing for this. This method can narrow the validation scope for more accurate experimental research and reduce resource consumption. By identifying neural circuits in the neural connectomes of C. elegans across different developmental stages, we analyze the developmental process of distinct neural circuits. Okay, here they get into community detection. Community detection is a pivotal concept in network science, aiming to identify node clusters within a specific network or within a complex network that exhibits dense internal connections while maintaining sparse connections with external nodes. So this is where you're getting these clusters that are kind of defined by sort of a self-containedness. So there are dense connections between those nodes and sparse connections with other nodes that aren't in the cluster. And as you can imagine, this takes a lot of effort to try to discover these and their extent, so the extent of a community in a network. You always have to have some sort of criterion for sparse connectivity and dense connectivity, and sometimes that's a pretty fine line, basically. These communities typically correspond to functional structures in both biological and artificial systems. So we can think of an example like a social circle in a social network, a neural circuit in a neural system, or local computer clusters on the internet. And so we can think about, things that, first of all, we can label and define those pretty easily. Those are things that we know what we're looking for. And so we do actually know in C. elegans connectome what we're looking for. We're looking, say, for maybe something associated with the motor circuit, or something associated with like a feeding circuit. And then we can then find the cells that connect to the main cells of that circuit, and we can define that as a community. So it's harder to do when you don't necessarily know what you're looking for. There are different modes of community discovery in networks as well, especially when you don't have a good characterization of what the functions are in your network or what cells are involved in a certain function. So it makes it harder in larger connectomes to do this. But people do community detection in, say, like the human connectome and systems like that. The emergence of communities and networks can generally be explained by the strength of weak ties theory. And this is a theory that goes back all the way back to Granovetter in 1973. This is the idea that you usually in networks find clusters. So you might find different friendship networks or social groups or functional groups within networks. And then there's some nodes that provide a link to other groups. So if you think about social groups, I mean, you have maybe a small social group, they're very tight knit, but there's one person that connects to another group. And so that other group also has a very strong membership criterion. But there's a link there that's weak. It's only based on one person, but it's persistent and it's critical to the functioning of the whole. So, being able to sort of have a community within a connectome allows you to have very specific types of functions, but also connect that to other types of functions in other parts of the connectome. So that's kind of what it's based on. It's based on this idea that you have these you have these very tight-knit groups, but you have these members of those tight-knit groups that connect in a sparse manner to other such groups. So the academic community has proposed various methods for network community detection, including modularity optimization, random walk, spectral clustering, and machine learning. So there are all these different algorithmic techniques that people use to find communities. They're all relatively computationally intensive. So if you have a very large network, they're very hard to implement. So this is another advantage of C. elegans, is that you can use a network that's 302 neurons in size at its maximum with maybe thousands of connections to explore. And that's a small enough size to really do these kinds of community searches thoroughly. These methods can be broadly characterized into two types. So we have two different types of community network detection: non-overlapping and overlapping. So non-overlapping, of course, is looking for communities that are distinct, in this case, distinct neurons that don't overlap with any other communities. So this is what we might call strict modularity of function. And then we have overlapping community detection methods, where you're looking for communities that are tightly knit and have those weak ties. But they have those weak ties in that they include nodes that are in maybe more than one community. And so, you can think about a community of cells that maybe are involved in motor control, but a couple of those cells, or maybe one cell, is also involved in the feeding circuit. And so, you can think about, the connections between those two functions and why those connections might exist. So this is, there are different ways in which these weak connections can be made. Non-overlapping methods identify structures where each node belongs exclusively to one community, whereas overlapping methods allow nodes to be simultaneously affiliated with multiple communities. So there is this aspect of labeling the cells and knowing kind of what their pre-existing knowledge of what their function is. So it is sort of a label technique of doing this. So again, if you don't know exactly what cells do, if you don't know the range of their functions, it makes it a little bit harder to do this kind of search accurately. But I think at the level of the C. elegans connectome, I don't think that's a huge problem, because you're not searching a very large space. The nervous system of C. elegans is a weighted and directed complex network consisting of 302 neurons and over 7,000 synaptic connections. So this, again, we have synaptic connections. We have the data from, this is from Cook, which is the data set where they look at the male connectome, and then also the Whitvliet data set where they look at the larval connectome. Within this network, numerous mutually overlapping neural circuits exist, with individual neurons belonging to multiple neural circuits simultaneously. So we have these overlapping communities. This overlapping architecture enhances the nervous system's flexibility by sharing neural resources across distinct functions, enabling it to perform multitasking and adapt to complex dynamic environments. In this study, we utilized a modified big clam algorithm, and that's cited by Yang and Laskavec in 2013. This is a method for overlapping community detection in weighted and directed networks to identify neural circuits in the C. elegans nervous system across different developmental stages. Okay, so then they have figure 1 here, which is the synaptic connection strengths of neurons of C. elegans across different developmental stages. So we have synaptic connection in strength, synaptic connection out strength, and synaptic connection strength. So we have, I guess, into the cells, out of the cells, and then the overall strength. And we can see that, basically they have a bunch of cells that they're analyzing, and then they're analyzing these rich club cells, which are, of course, we've talked about the rich club cells, where those are the highly connected cells in the connectome. And we see RIAR and RIAL, so the left-right pair of RIA, are strong both in the in-strength and out-strength categories. And then the rest of your rich nodes are typically leading in this, in this ranking. And the red line is the average, so they're above average. And then in the synaptic connection strength overall, RIAR and RIAL are also very much distinctly have a stronger set of connections than all the other rich nodes, rich club nodes, and then all the other nodes, period. So that's what these graphs show. So for each graph, so for graph A, which is in strength, synaptic connection in strength calculated exclusively based on the count of incoming synapses is represented here. So this is things coming into the cells. This is the strength coming into these cells. Synaptic connection out strength calculated exclusively based on the count of outgoing synapses is representative of B. So this is where they're counting synapses. They're then making a proxy. That's a proxy for connection strength, and then that's how we calculate that. So the RIA cells are receiving a lot more synapses, both in and out, than any other cells in the connectome. And then synaptic connection strength, calculated based on the total count of synapses associated with the neuron, is in C. That's obviously going to be the case because A and B basically show the same information, just in two different parts. All right. So again, they use this Whitvliet data set, which is, of course, where we have data at different stages, L1 through L4. We actually have 8 data points here. We have 8 isogenic individuals encompassing multiple developmental stages, specifically 4 individuals at the L1 stage, 1 at the L2 stage, 1 at the L3 stage, and 2 at the adult stage. So we have the different time points, and they're looking at these different points and seeing how this connectome varies across those. While the prior research conducted statistical analyses on multiple metrics included in these neural connectomes, in the present study we only selected the synaptic count connectivity matrices which is the presynaptic neuron times the postsynaptic neuron, which gives us 180 times 224. So this is where we're, sort of creating a specific subset of the data that encompass all neurons, muscle cells, and glial cells as a core analytical data. So this is figure 2. This is 2 viewpoints on the structure of overlapping communities and complex networks. A, which shows overlapping networks, or I guess overlapping communities in a network. So you have one group here in green and another group here in dark red. And then this mutual set of connections here with a bright red color, denoted with bright red color. And those are technically members of both communities, but they overlap in terms of function. And so in the connectivity matrix, we should expect to see this pattern here, where we have dense connectivity in the connection matrix for both groups, but a sparse region here, which denotes this overlap. That's one scenario. This scenario here shows a similar sort of situation, but instead of having sparsity in the overlap, we have sparsity in the areas that don't actually sparsity in the areas here that don't represent either group, and then a lot denser connectivity in this overlapping region. And so if we take these two examples, then we see kind of what's going on here. We have the neural circuits A and B. They're distributing to these different nodes, and A and B is distributing or contributing a little bit to each of nodes D and E, which are the overlap. And then, of course, with our neural circuits A and B, then they come down to this neuron U, and then you can partition the contribution of these two connections as FUA and FUB. So that's kind of the contribution of each component to that neuron. So, yeah. And then each affiliation edge from neuron U to neural circuit C has a non-negative weight if FUC greater than 0, indicating the strength of neurons U's belonging to neural circuit C. So this is, this U down here is this C. This is a new group or community C where you're getting contributions from A and B. So this is figure 3. These are the statistical results of identified neural circuits across all developmental stages. In A, which is on the left, the proportion of identified neurons relative to the total number of neurons across developmental stages is shown. So we have our different stages L1 through L4, and then we have adult as sort of a control. And then we have a proportion of coverage. So it's pretty complete at L1. So we have 85% proportion of coverage, and then that goes up to 96% as the adult. So we have some fluctuation between L1 and L3. L4 is basically identical to the adult. In B, we talk about the number of neurons, and again, this is divided up by developmental stage. We have these plots where it shows the average value is this dot in these each of these red blobs, and the red blobs show the variance. So you have the number of neurons which are variable from L1 to L4, then to the adult, and this just shows the profile of these cells. So L1 and L2, you get a lot of variance skewing up to the, a high number of neurons. L3, it's kind of more concentrated or contained. L4 and adult then have the stretched out pattern that you see here with some variation between L4 and adult. And then the number of connections per cell varies again, L1 through L3, and then L4 in adult. So to give us an idea of what these changes look like in the aggregate. So L1 through L3 is where a lot of things are happening in terms of post-embryonic morphogenesis. You're getting a lot of seam cells born at that time. You're getting a lot of changes in connectivity due to environmental factors. There are also different types of diapause that the worms go through. So in L1, you can actually induce a starvation stage of development where you can hatch eggs in an environment with very little food, and you can get them to survive or persist in that L1 phase for a lot longer than they would normally be in that phase. You can arrest development and then put them back on food, and that affects their metabolism and their growth. Similarly, from L2 to L4, we have this dower phase, which is where there are a lot of physiological changes associated with persistence in starvation conditions. So they grow this really thick cuticle, and there are other changes that happen as a result of this going into this dower stage. And that, of course, is going to, it's sort of triggered in L2 and it persists to L4. So we see a lot of the plasticity happening in L1 through L3. And that's consistent with what we find in a lot of the other work. This is figure 4, neural circuits spanning L1 to adult. So now we get into the neural circuits component here, where we're looking at these networks with labels. So A is here. This is a series of network motifs that go from L1 to L3. So you have your cells that are alive or in this part of the network at these different times. So we have, I guess this is how it's set up. We have connectivity changes, and I don't know what the gray cells are for. I don't really explain this very well here. But basically, okay, so A is the developmental trajectory of neural circuit mediating pharyngeal regulation. So this is a pharyngeal regulation circuit. These are the neurons involved in this at L1. Then it changes its connectivity slightly in L2. It drops out ADA and it picks up URY. And then in L3, it's picking up BDU and it's changing its connectivity as well. And it has these grayed out cells kind of in the periphery. So IL2, RIH, and URY for L1, and then IL2, RIH for L2, and then IL2, RIH, URY, and PVR for L3. So PVR is a previous component of the circuit that drops out in L3. The same with URY. RIH is persistently out of the circuit, as well as IL2. I guess this is what it looks like in the adult, so that's why those cells are there. Okay, yeah, so that's what happens here. So now we get to L4 in adult. So in L4, then we establish these connections. But here in L4, we start to establish this rich club connectivity, which is a higher level of connectivity between IL2 and RAP. And then we connect them all together in the adult. So that's what this is. So the gray ones are the ones that drop out or are present in the adult circuit. And you see the changes over time. So you see where URY becomes connected and then it loses its connection again, and then it becomes connected again by the time we get to adulthood. B is the developmental trajectory of the neural circuit mediating protected sleep. So B is the developmental trajectory of the neural circuit mediating protective sleep. So this is a different function, different neurons. Actually, there's some of the same neurons that are overlapping here. URY is one of them. CEP is another one. So we're actually using, this is actually maybe like an example of the weak ties between pharyngeal regulation and protective sleep. So that's what we have in B. And then again, we have the same thing where we go from L1 to L2 to L3. In L1, we have a very small network, CEP, OLL, RMD. Then in L2, we start to get this rich connectivity pattern, as well as most of our cells connected here. Only URY is out of this stage circuit. And then in L3, we drop RMD back out. And then in L4, everything is connected as it would be in the adult. And then I don't think there are any changes between the adult and L4. So everything is in the network at L4, and then it persists to adulthood. Now you can see that there are these different categories: sensory neuron, interneuron, motor neuron. So, again, you can look at these different classes of neuron. So it's not just the label that we have labeled, or the single cells we have labels for. It's also their overarching function or their overarching type of cell. So motor neurons express certain things, interneurons express other things, sensory neurons express other things. So they're always involved in different types of stages of the sensory motor process. So sensory neurons are taking in sensory information from the environment. Interneurons are connecting sensory and motor neurons, and then motor neurons are providing an output to muscle. So that's, and again, they're expressing different things, and we would expect them to behave a little bit differently in this network. And we also expect the network connectivity to vary depending on whether something is a motor neuron or a sensory neuron or an interneuron. So if we look at it in that light, we can see that like PVR dropping out in L3, but then coming back online in the adult, that means something. And, we can follow up in terms of what PVR is doing specifically, but also why you would disrupt the interneurons in this network sort of temporarily in this way. So it turns out that PVR is sort of on the periphery here, and it's serving some functions. So, we can follow back up on that if we would like. And that's the advantage of this approach, is that it gives us this dimension of development, of wiring during development, but also of changes in function and what the effects of those are. Okay, so then they explain pharyngeal regulation. So what that is, so in wild-type animals, gentle touch detected by extrapharyngeal mechanosensory neurons transiently suppress pharyngeal pumping, an effect abolished following RIP ablation. So if you take out the RIP cell, which is in the middle of this community, you can basically destroy the function. Because as you see, it's right in the middle of the action. It has a lot of connections coming in and out of it. All right, so that's, something we can observe in, if we do this experiment in adult worms, we can get rid of that one cell and we can get rid of the function. But other cells in the network, of course, if we ablate them, we only sort of suppress the function a little bit. And, taking these out of the network can change the pumping action. CEP and IL-1 are two classes of head-localized mechanosensory neurons. CEP enhance feeding efficiency in the presence of food by regulating the basal slowing response, whereas IL-1 neurons primarily modulate spontaneous foraging movements. Furthermore, both CEP and IL-1 respond to head touch and subsequently direct locomotion. So just describing what these different neurons do in the adult. Some neurons traditionally categorized as interneurons or motor neurons also exhibit sensory capabilities. Among these neurons, PVR is a tail-localized interneuron, with neurites projecting posteriorly into the tail spike and anterior projections extending along the right lateral cord to the ventral bundle, where it forms synapses onto RIP, so that neuron that we talked about. So given this anatomical profile, PVR may transmit tail mechanosensory signals to the pharyngeal neural circuit. RIP neurites further innervate neuromuscular junctions formed by sensory neurons IL-1 and URA, which points to a potential functional crosstalk between IL-1, URA, and RIP. So we can establish all these functions and what different neurons are doing and what their connections are doing. And then we can do these manipulations or look at how they kind of come online and maybe go offline transiently in development. Okay, so these are two other functions here. So this is A is, and follows the same convention as the last figure. So A is the developmental trajectory of neural circuits underlying multisensory integration. So this is multisensory integration where we're integrating, say, like touch and chemosensation. And this is, of course, important in foraging so that they can find things to eat or sense their environment. So here we have a lot more things that kind of are offline in L2. We have a small circuit here of 5 neurons. And then the configuration changes significantly in L3, not just in terms of the cells, but in terms of the constituents of that network. So we have sensory neurons and motor neurons exclusively. Then in L4, we have this, we add more cells into this network. And then in the adult, we have this full network with this rich club connectivity. Okay, so that's multisensory integration. And the thing about multisensory integration is you need to have sensory neurons really to make this work. We have sensory neurons, throughout. So in L3, we have, what did I say, sensory neurons and motor neurons exclusively. We don't have any interneurons. Whereas in L2, we have one interneuron. So there's something that, the interneuron drops out for some reason. And then in L4, we gain interneurons back in. The second one, of course, is this developmental trajectory of neural circuits mediating egg-laying aversive stimuli. So this is specific to egg-laying and whether worms should lay eggs or not. And what's interesting about this circuit is now we have one neuron, HSN, which is a sex-specific neuron. So this is where HSN is in worms that lay eggs. And of course, 99% of worms are hermaphrodites, or sometimes 100% depending on the population you're looking at. So HSN is not expressed in males. They don't have HSN, so they don't have a functional form of the circuit. But anyways, we can see that this network has a similar pattern of coming online. In L2 and L3, we have interneurons and sensory neurons. And then we finally add in, we don't have any motor neurons in the circuit. Then we finally add in our sex-specific neuron in L4 and in adult. So we can see that this network sort of is generic with respect to sex specificity until L4, where the HSN is born and is connected into it. And of course, there's no need to lay eggs until L4. So having this here at L4 is fine for the function. And this is actually, when you get, when males transform from hermaphrodites, which happens during this period, it's usually during L4 when they start getting this tail spike, which is a definitive of the male phenotype. So that all makes sense. So then finally, this figure 6, let's go over this one. This is a neural network. This is a neural circuit spanning L3 to adult. So the first figure was L1 to adult. The second one was L2 to adult. And this one is L3 to adult. And this is for oxygen-related egg laying, so a neural circuit involving that in A. And then in B, we have a neural circuit for mechanosensory motor control in the anterior head region. So we're getting more specific in terms of the functions. And so this first one, which is oxygen-related egg laying, we have something that's exclusively interneurons in L3. Then we gain a sensory neuron in L4, along with a sex-specific neuron in L4, which is also HSN. And then in the adult, we add in another interneuron. So this is one sensory neuron, a bunch of interneurons, and a sex-specific neuron. And the sensory neuron comes into play in L4. And again, we don't need any sort of sex specificity or any egg-laying behaviors before L4, so this kind of makes sense. Then this final one here, which is mechanosensory and motor control in the anterior head region, involves sensory neurons, motor neurons, and one interneuron in L3. Everything is connected in L4, so we gain GLR, which is a motor neuron, and then in the adult, we basically gain some connectivity. So that's what we're doing here. So this is a lot more, so it starts out a lot better formed than some of the other networks, and this happens in L3, and then it proceeds to L4 in the adult. All right, so that's that paper. I thought that was a very interesting paper. They did a lot of good work in that paper. It's getting deep in the weeds in some of these behavioral circuits. I did want to talk about two other papers here really briefly. This one is mapping morphogenesis and mechanics in embryo models. So this paper is from 2023, but I thought it was interesting given some of the things we've talked about in the group. This is Zhongwan Liu, Vikas Trivedi, and Idse Heemskirk. So this is where they're looking at geometry and forces in the early embryo. So recent methodological advances in measurement of geometry and forces in the early embryo and its models are enabling a deeper understanding of the complex interplay of genetics, mechanics, and geometry during development. So over the last decade, in vitro stem cell models have provided a powerful approach to understanding the underlying principles of early mammalian development. So we're talking about mammals here and mammalian systems. This embryoid revolution was founded on two key insights. First, control over the initial geometry and cell number is crucial for self-organized patterning. And second, interactions between embryonic and extraembryonic tissues is required for morphogenesis of structures resembling the early embryo. Therefore, we find it useful to classify stem cell models according to their initial cell configuration, which then determines their complexity and the developmental processes they recapitulate. The simplest systems involve only pluripotent stem cells that contribute to the embryo proper in vitro. Gastroloids are used to model gastrulation, the stage of embryonic development where the body plan is laid out and the three main lineages of the body, ectoderm, mesoderm, and endoderm, are established. Gastroloids come in several variants, displaying different degrees of patterning and morphogenesis. 2D micropattern gastroloids start from a disc-shaped epithelium of PSCs or pluripotent stem cells on a substrate. These initially form highly reproducible cell-fate patterns with limited rearrangement, but later undergo morphogenesis in a more complex multi-layered structure. In contrast, 3D gastroloids, so we have 2D gastroloids which are on a plate, usually a cell culture plate, and then 3D gastroloids which are usually in some sort of 3D culture or bioreactor. The 3D gastroloids start from a spherical aggregate of PSCs that undergo spontaneous symmetry breaking, accompanied by extensive cell rearrangement and subsequent axial elongation. Other variants start with an epithelial cyst that is a hollow sphere that resembles the disc-shaped model after spontaneous or explicit symmetry breaking. So for them, symmetry breaking is important in the geometry. Adding an initial differentiation step towards a particular lineage allows aspects of post-gastrulation development to be recapitulated with similar approaches. So, for example, parts of the ectodermal patterning and morphogenesis can be recapitulated starting from an ectodermal disc and cyst, respectively. More complex models combine embryonic and extraembryonic cell types. So these are like blastoids, which combine small numbers of extraembryonic trophoblast stem cells and embryonic PSCs, which form a 3D spherical structure resembling the blastocyst. So these are all approximations of these different structures in embryonic development. Mouse embryoids embed these cell types in extracellular matrix, while ETX embryoids include extraembryonic endoderm. In both cases, a cylindrical epithelial structure resembling the mouse embryo at the onset of gastrulation arises. ETIX embryos include extraembryonic endoderm-like cells induced by transcription factor overexpression and can develop much further, completing gastrulation and continuing into organogenesis. In our opinion, each stem cell model should be viewed as simplifying development in specific ways that enable answering questions that are difficult to address in the embryo. So differences in vivo development can be useful features rather than shortcomings. By comparing different models and leveraging their individual technical advantages, developmental mechanisms can be revealed that are obscured by the complexity of the full embryo. So, for example, embryo models have demonstrated how patterning of embryonic tissue occurs in the absence of morphogenesis or extraembryonic tissues, and have provided insight into patterning mechanisms by varying tissue size and shape. So they believe that the next challenge lies in understanding how patterning, mechanics, and morphogenesis are coupled. Certainly, embryo models will play a key role in this by enabling comparison of different tissue configurations. Here we describe the recent advances in methods for understanding the disparate spatial organizations of embryo models at different scales, with an emphasis on how tissue flow and geometry can be used to infer tissue mechanics. Okay, so they get into how we relate flows and forces in embryogenesis. And so they point out that morphogenesis can be described very generally by tissue flow. And they define tissue flow as a local rate of displacement. So it's the flow of tissue or the rate of displacement across that tissue. Tissue flow can be obtained by imaging data directly at the pixel level using particle image velocimetry, which is PIV, or optical flow. And Susan knows a little bit about this. In a first application of such techniques to embryo models, optical flow was used to demonstrate that the endodermal cap forms by collective cell migration in 3D gastruloids. Alternatively, tissue flow can be obtained by cell tracking, which provides more information about the cellular level movements that underlie the overall tissue shape change. Recent improvements to cell tracking include machine learning approaches, and there are different ways we can optimize that. And a lot of automated cell tracking techniques that allow us to automatically discriminate spatiotemporal features of cell lineages and cell division events. This is stuff we've been doing with some of the data sets where we're trying to find the lineage tree and division events within images and things like that. So these methods were shown to be very effective at analyzing mouse gastrulation and can be anticipated to soon find application in studying embryo models. So then they get to tissue flow, which is generated by mechanical forces, with the relationship between flow and forces depending on the mechanical properties of the tissue. The tissue can generally be considered as an active viscoelastic material, meaning that it has both fluid and solid-like properties and is therefore characterized by both viscosity and elasticity. So we can use Hooke's law to relate the deformation of a spring to the applied force given the spring constant. And we can look at tissue flow in chick gastrulation to see how it behaves, and indeed it behaves as a viscous flow driven by a ring of tension on the embryo margin that is not simply downstream of embryonic patterning, but is part of a coupled mechanochemical patterning system. Similar applications can be expected for embryoids in the future. Okay. And so force inference from cell and tissue geometry is powerful because it is global and high throughput, but rests on the assumption about the mechanical properties of tissue and only provides the relative forces. So, we can try other techniques like using fluorescent force sensors, but there are problems with those kinds of methods. So there are other methods that are up and coming that allow us to look at absolute mechanical stresses over time as a result of deformation of injected liquid droplets or gel beads with known mechanical properties. So this is, these are going to be useful in determining how the mechanical properties of three-dimensional gastroloids change as they differentiate. So the thing about like embryos and embryoids is that they're these large aggregates, especially embryoids where, you have to track a bunch of things at once. So they use coarse-grained approaches for this. So one thing about embryo models is you can produce thousands of structures per experiment, but of course, being able to measure the tissue flow and forces in every cell requires really high resolution imaging at both the spatio and temporal scale. So we can't really do this for things like embryo models or 3D gastroloids. We need a coarser grained description for shape. And so for this purpose, methods were recently developed to capture large-scale features of the geometry such as volume, area, eccentricity, or number of lobes from what are basically low-resolution bright field images. So that's what they're getting at here. They talk about tissue layers and epithelial morphogenesis, which is that some of these high-throughput methods, although they're very good at capturing large amounts of data, just basically outline structures in bright field images and thus contain limited information. So when we do a segmentation of an object in a bright field image, we're just basically selecting that feature and then counting it as that feature, or we count some aggregate property of that feature. We oftentimes don't drill down and detect things like the membrane or the components of the cell. So this is a problem of granularity, but it's also a problem of, volume, because you can't really do that when you're dealing with thousands and thousands of images. And so, we need high resolution measurements, and specifically of tissue flow and in 3 dimensions to really make this work. But even when you do have those, they're hard to interpret. So we need to have new methods for this. We need to have methods that kind of bridge that gap between high resolution and high throughput. And so they talk about getting into tissue cartography and this method called tubular, which is tub and then ular in capital letters, a method to globally analyze deformation of complex dynamic tissue geometries. And so tubular establishes a material frame of reference in which cell movement is minimized and decomposes tissue flow into different in-plane and out-of-plane components, greatly simplifying dissection of epithelial morphogenesis into its constituent cell behaviors, such as rearrangement, shape change, and proliferation. So being able to take all of the cells and their movements and then figure out the different components of the images, and then being able to kind of extract different types of constructs that tell us something about the physics and the geometry. So the focus here is on tissue cartography and thinking about tissues like a map and the properties of that map. When we have a map, we usually have different properties on the map. We have different levels of detail. We have different things happening on the map. The map is usually a static representation of a place or a region. And so, of course, things happen in those regions that aren't represented on a map. Dynamic things like migrations or flows and transports of liquids or other types of things, deformations over time. And so we can actually extract those. We can represent those on a map. If we have the information, we can extract that information if we have maps from multiple time points. So that's kind of the approach they're taking here.
I just briefly want to say I have a small gastroloid. It's called a salamander egg.
Yeah.
So that's basically, yeah.
Any insights on that, or like, how are you measuring? Yeah,
You measure it with a light microscope
yeah,
and you can see movement. And if you get imaging from enough angles, you can get the whole gastroloid egg.
yeah,
Yeah. So that's what I've been trying to do.
yeah,
But I think the blastocele sets up the whole body axis. So I would like sort of a baseline. Like I'm hoping this student will just take images over time, and you can watch the interior blastocele grow, because it does put pressure on the cells. Like you can see that they're kind of rounded when it starts to develop, and then as it progresses, it becomes flattened, like the cell structure is flattened.
yeah.
Also say Natalie Gordon is an expert on some of this,
Oh,
if
yeah,
anybody's interested.
yeah.
Anyways, yeah, and if you want to know anything about or some summary of what Vic's lab looked like in 2004 to 2000 and when was it, 8 or so,
Yeah.
I was there.
Okay.
But had salamanders, like he had these big tanks with salamanders in them, and he tried to keep them in kind of a natural environment.
Right.
So yeah, so it was an interesting lab.
Yeah. Yeah.
And yeah, and then they decided that the animals had to be in with the animal control people, and and it was a mouse lab, and so it was too hot for the salamanders.
Yeah.
It was too hot, and yeah, there. So then they put in a air conditioner to cool it down, and it heated it up.
Oh, yeah.
So when I heard that, I burst out laughing, because it's 101. You put a refrigerator in an insulated room, and is the room hotter when the fridge is open or when it's closed? It's a trick question.
Yeah.
It's hotter when the fridge is open,
Okay.
because it's using more energy.
Yeah.
So what the pipe going out from this air conditioner was heating the room up. So I said, well, put insulation on the pipe. they had a, yeah.
Yeah.
And then they didn't like it because it was pink insulation. So they took it off right before a long weekend. And so the poor technician had to come in and put ice in the water. And then all of the salamanders all of a sudden laid eggs.
Oh, wow.
So now I can get my salamanders to lay eggs.
Oh, good.
You upset them a bit, and then you put cold water on them. Put a male and female together, you upset them a bit, and then put cold water on them or ice. They will lay eggs.
Oh, wow.
So that's how you get axolotls to lay eggs.
Yeah.
If anybody wants to know.
Yeah.
Probably not.
All right.
Anyway, yeah.
So yeah, this paper that I'm gonna finish up here soon. I think we're through most of it. Then they talk about small aggregates and early embryoids. So before forming large epithelial structures, embryo models typically combine small numbers of embryonic and extraembryonic cells in suspension. The internal dynamics of these cells as they rearrange and organize structures is of great interest. Then recent advances were made for subcellular segmentation of small embryos. including mouse and ascidian embryos, with the goal of more accurate inference of mechanics. And then there are different types of segmentation techniques, and that are appropriate for this type of complex three-dimensional image. The types of images you need to sort of analyze to get a good account of these structures. Then, so the outlook is that we should look at the interplay of patterning and morphogenesis. So traditionally, tissue patterning and morphogenesis have largely been studied in separation. However, it is now clear that these processes are intricately linked through an interplay between mechanics, cell signaling, and gene expression within a dynamically changing geometry. Recent methodological advances have substantially improved our ability to relate mechanics to tissue geometry and embryoid models. One limitation of current methods is their inability to address interactions between tissue layers, and we view this as an important future direction. For example, epithelial force inference methods have focused on apical force balance, and it will be an important factor to determine how these are coupled to basal forces of the underlying extracellular matrix and other tissues. So, for example, in the case of the epiblast and hypoblast, we see this sort of arrangement where we're looking at apical force balance and how that's coupled to basal forces. Furthermore, limited attention has been paid to the mechanical interaction between the mesenchymal cell populations arising in gastrulation and the surrounding epithelia. As our toolkit expands, we believe the comparison between real embryos and stem cell models that decouple morphogenesis and patterning to varying degrees will enable confrontation of the complexity of this interplay, both in vitro in gastruloids and embryoids, and in vivo in actual embryos. So that's that paper. All right, looks like Barshan had to leave. Did you have any more comments on that, Susan?
No, not really, but I'll look at it. It relates to what I'm trying to do.
Okay, sounds good.
To look at the light field images of salamander eggs, and as they're developing their early brain, like the neural plate and then the brain. So I'll get images of it eventually. I've got 8 youngsters in my tanks at the moment. I've got 8 teenage axolotls. So right about maybe November, they will, or October even, they can lay eggs. I could probably, well, it's not good to ask them to lay eggs at this time of year, but they might, because they're teenage axolotls, and they work better for eggs.
Okay. Yeah.
Yeah.
Yeah. So this is the final one today. I'm not going to go over this in too much detail, but this is an interesting paper nonetheless. This is from Physical Review X, and this is brand new. Arvind Murugan, David Zwicker, Charlotta Lorenz, and Eric Dufresne are the authors on this. And again, it's another paper where there are materials scientists and physicists involved in this. So the paper title was "Could Living Cells Use Phase Transitions to Process Information?" So I think if you're a fan of physics or biophysics, the answer might be yes, and you might have a very good idea of how that works. But they're going to look at this question sort of in the context of homeostasis. So let's read the abstract. So to maintain homeostasis, living cells process information with networks of interacting molecules. Traditional models for cellular information processing have focused on networks of chemical reactions between molecules, So this is defining cellular information processing as these networks of chemical reactions. We just talked about networks of neurons and how those are connected together and how those result in function. And of course, now we're talking about chemical reaction networks, which are different, of course, because they have different ways of, being connected and staying connected. But in any case, you're looking at these kinds of physical interactions. And so that's what's important here. So here we describe how networks of physical interactions could contribute to the processing of information inside cells. In particular, we focus on the impact of biomolecular condensation. So we've talked about condensates in the past, and it's kind of a hot topic in biological physics and in molecular biology, that we have these condensates of things in the cell membrane or in the cell cytoplasm, and we have, they're just kind of aggregates of different biomolecules. And people have, there's this history of people wondering what exactly they did there. If they were just kind of leftover junk, or if they were kind of, functional. And if so, what is their function? Are they key to cellular function, or are they just there because, we have a lot of things in the cytoplasm? And it turns out that they have a number of interesting functions. And, people are still kind of trying to unpack that. So they focus on the impact of biomolecular condensation, which is a type of structural phase transition found in cells. So this is something where they actually serve not only as a place for concentrations of molecules, but it also provides a structural phase transition. Biomolecular condensation has recently been implicated in diverse cellular processes. Some of these are essentially computational, including classification and control tasks. We place these findings in the broader context of embodied computation, an emerging framework for describing how the native dynamics of nonlinear physical systems can be leveraged to perform complex computations. The synthesis of these ideas raises questions about expressivity, and that's defined as the range of problems that cellular phase transitions might be able to solve. So if you're familiar with the topic of expressivity and graph neural networks, it's a key area. And it's just thinking about what can you solve with this method. And the same thing holds true here. So we want to know about the expressivity of condensates, but also the learning aspects of condensates, or how these systems could adapt and evolve to solve different problems. So this emerging area of research presents diverse opportunities across molecular physics, soft matter, and embodied computation.
So would that include epithelial to mesenchyme transitions, I suppose?
I guess so. Yeah, it might. I haven't read. I mean, I guess they're thinking very broadly about this, so it might be something like that.
Okay, because it'd be interesting to know if condensates were involved.
Yeah. That'd be interesting.
I always thought it was maybe because their junctions broke.
Okay.
And that's why it changed, but I don't know how that happens.
Right.
That switch. So yeah, be interesting to know.
Yeah. So again, they focus on thermodynamic equilibrium and homeostasis. So living systems are usually far from thermodynamic equilibrium. They achieve a healthy steady state or homeostasis across length scales through energy-consuming closed-loop control. So they have this control imperative. They have to maintain their, a healthy steady state, and they need things to help them to do that. So there's the idea here is that there are these computations going on in the cell that kind of maintain this homeostasis, because the cell, of course, is very far from thermodynamic equilibrium. There are always fluxes happening in the cell. Proteins are being made, genes are being transcribed. You have things coming in and out of the cell, and their interactions with the cell and other cells. So there are all these things that are kind of pushing and pulling on the cell's homeostatic state. And so these computations have to be extensive, and they have to be persistent. Even an individual cell can sense and compute to mount an appropriate response to external stimuli or internal changes of state. And they show this in figure 1. And so there are all sorts of environmental cues, internal cues, and other types of things. From an information perspective, homeostasis is a control problem. So in some cases, cells respond to relatively simple one-dimensional stimuli, such as the concentration of a single chemical species. So E. coli, for example, or neutrophils move themselves up a concentration gradient in a process called chemotaxis. Cells also process a vast array of physical and chemical signals, so this could be matrix stiffness or responding to soluble matrix-bound growth factors. Processing these high-dimensional stimuli can include classification, the assignment of a label to a broad range of input values to summarize the relative pattern in the signal. So cells do a form of classification to respond to chemical signals in its environment. It's just where they pick up different signals and they mount a response. And they have to, understand it. They don't really have to understand anything. They just need to be able to put it in the right category and mount the right response for it to continue to be in homeostasis. So the stakes are pretty high, but the sort of discrimination there is relatively low. Classification allows cells, okay. And so, we need to do this for things like advancing the cell cycle, switching off mechanical state, or differentiating into specific cell types. So again, classification has been recognized as an essential step in stem cell differentiation and other things. And so this is where, particular differentiation pathways require discrimination between many ligand combinations and temporal patterns. So in this perspective, we discuss how two emerging topics in physics, biomolecular condensates and embodied computation, could complement each other and provide fresh insights and new tools for information processing biomolecular networks, particularly those inside living cells. And then they kind of get into the basic physics of condensates and how they could automatically achieve certain computational tasks by classification and control. We then generalize these behaviors using essential ideas of embodied computation and highlight open research questions. So I will stop here for this week, and I may revisit this paper next week. We can go into it in more detail. But that is, those are 3 papers I think are worth reading more, especially the last one, because it's quite interesting with respect to maybe even the second paper, because the second paper talks about, the geometry of the embryo and what's happening at sort of the global scale. And this is talking about information processing in single cells and then bridging that gap. What can we learn? You're muted, Susan.
And there we go. Thank you for those papers. Yeah, I'll try to find time to look at them.
Yeah.
Yeah, because they certainly relate to what I'm doing.
Yeah. All right. Well, I think that's all for today. Thank you again for attending, and see you next week.
Okay, bye.
Bye. Thank you.